This video made possible by Killer Visual Strategies. Visit killervisualstrategies.com.
Thank you. Is this on? You guys hear me okay? Great. So thanks to the GeekWire organizers for the opportunity to be here today and talk to you about our work. I'm going to give you guys a lot of things to think about in the next 15 minutes. If you want to have a continued conversation, I'd also like to give a plug that I am one of the 12 who will be joining Luke Timmerman and his trek to the Everest Base Camp next spring, and we still have 8 slots if anyone's interested. So I'm going to be talking to you today about gene editing. And since this is GeekWire, I thought I'd make a computer metaphor out of it. So if you imagine that each human being on the planet is like a custom computer code. We all have similar code that is telling our bodies to have specific operations, things like eyes, hair, skin, lungs, blood. But the exact codes that dictate those functions or those operations are unique to each individual. Small changes in those codes are things that we've developed over evolution. It's what makes every one of us in the room look different. Sometimes they're superficial, like the eye color shown in this picture, but other times they can have detrimental effects on the function of our operations. And one particular that I'm going to focus on is sickle cell disease, which is highlighted here. This is a single line of code that has an error in it. And that error causes a compromise in the function of red blood cells, which we need to carry oxygen and nutrients all over our body. This genetic code error causes the red blood cell to disform. That causes clogs in the blood vasculature, extreme episodes of pain, a higher likelihood of stroke, and organ failure, and leads to an incredible healthcare burden for the individuals. And there are tens of millions of them on the planet who are impacted by this disease. So as long as we've known what our code is, DNA, since Watson and Crick discovered it, which was before I was born, we have postulated that genetic editing could be used to correct those errors in our human code. Many people, for lots of decades, have been working on different tools to perform these corrections, but the biggest revolution just happened in the last decade, and it is called CRISPR. Has anyone in here not heard of CRISPR before? That's all right, I'm going to give you a brief explanation. So, as I mentioned, there are many tools that people have been working on. This one has garnered so much attention because it was actually designed by nature. CRISPR is a system that bacteria developed to correct errors in their own code. When scientists discovered this, they realized that nature had sort of put together a very simple and elegant system that could possibly be translated to make corrections to the human code. So what is CRISPR exactly? Well, it's technically a very simple 3-part system that I'm going to illustrate in this cartoon. There are 3 components. The first I'm going to say is that hand there. The first step in this process is that the hand has to scan the DNA to find the place in the code where the error exists. The second piece is those scissors. The hand uses the scissors to make a cut in the DNA so that it can change the code there. And then the third piece is adding the critical code that needs to replace that error-prone code. Pretty simple system. When scientists discovered this, it was such a revolution that CRISPR now has its own scientific journal. Can you guys imagine if, like, next year's antiviral software had its own publication? It'd be pretty funny. But why don't we have access to this kind of treatment in clinics everywhere already? The answer is that it's not so simple to get CRISPR, these 3 simple pieces, into cells to perform genetic editing. So the first clinical trials are going on right now. They're actually to treat sickle cell disease. This is a disease of the blood, but that was a specific target because blood is something that we can take out of the body, manipulate outside, and put back. So we're not actually trying to deliver these things into people where there could pose more risks to other tissues in the body. But this process is complicated. Red blood cells don't live that long. Who here has donated blood before? Fair number of people. They tell you got to wait 60 days before you can come back and donate again. That's how long it takes for your body to replenish what you gave up. And the cells that are responsible for that are blood stem cells that reside in your bone marrow. If we could genetically edit or fix the error in the code for sickle cell in a blood stem cell for a person, that would be a one-time treatment that for their lifetime would prevent them from having the impact of sickle cell disease. It's a big step forward for this particular type of healthcare problem. But what it actually takes to do that right now is a patient comes to the clinic. Now, I mentioned these blood stem cells are in our bone marrow. They're very rare. They're very diffuse. They're spread out. We can't possibly take all of the bone marrow out of a person's body at one time. We have a couple tricks we could do to move those cells into circulation, but either way, it's a trip to the clinic. You're either going to the OR, or you're going to be on a machine for a couple of hours, possibly for as many as 3 days in a row. Once we've got that, we still have a blood product where the cells we want are very rare. So in order to get those cells, we actually have to do a complex set of preparations and use sophisticated technology to purify them away from all of the other cells in the blood or the bone marrow. Now we've got our cells and we're ready to add our CRISPR. But guess what happens when you put those three things, the hand, the scissors, and the new custom code in with these cells? Anybody got a guess? Errors. Nothing. Because cells don't want to take up bacterial pieces. That's probably a good thing, right? So the way that scientists have figured out how to get these pieces in is equivalent to someone jumping out at you from behind a door and scaring you, right? You go, right? They literally perform an electric shock of the cells. This causes them to temporarily open their membranes, and these things flood in. Now, this is great for getting the hand and the scissors in. It's not so great for delivering the custom piece of code. Cells don't like it very much, especially blood stem cells. So scientists said, well, now we need another way to get that custom code in. How should we do that? They turned again to nature and realized that nature has already found something that can deliver custom code really elegantly to cells, and that is viruses. So scientists are using this combination of electric shock and engineered viruses to get those three pieces in. Now, this is a lot of manipulation outside the body. We have to perform a lot of testing to make sure the cells are safe to go back into the patients. And then and only then can they go back to the clinic and be reinfused. In addition, we've done so much to these cells outside the body, and as I mentioned, we could never collect all of them. So cells that stayed inside the patient will always have an advantage. Thus, we actually have to treat these patients with chemotherapy, radiation, or other agents in order for these cells that we've genetically manipulated to be taken up. Now, this complexity and difficulty is partly to blame for the exorbitant costs that are associated with gene therapy, which I'll talk about in just another slide. I actually wanted to show this example. This is the facility where these kind of processes take place just down the street at the Hutchinson Cancer Research Center. There's a unique piece of equipment for every patient we're treating because we can't have two different patients' cells in the same piece of equipment at the same time. Nobody would want to run the risk of accidentally getting someone else's cells in this process. There are three people in this photograph, highly trained staff. One of them is reading instructions line by line. A second one is performing those instructions as they're being read. And a third person's verifying that the second person did what the first person asked. And all three of them are signing off that that's what was done. Multi-million-dollar infrastructure. Now, as I alluded to just a moment ago, these are the reasons that the current FDA-approved gene therapies have these really high costs associated with them, with Zolgensma being the winner, just approved last year. This is a big problem for diseases like sickle cell, because on a geographic scale, the patients with sickle cell, shown in blue here, don't live in places where we have these kinds of funds available for healthcare. In Nigeria, more children are born every year with sickle cell disease than in the rest of the world combined, and the average middle-class family income is equivalent to $500 US per month. The annual health expenditure per person in Nigeria is about $80 US. Now, if that weren't a big enough problem, I'm going to overlay on this map the places where not specifically gene editing for sickle cell disease is happening, but just that have the kinds of facilities I showed you in a picture on the previous slide. And if you don't see a big distribution gap, then maybe I didn't make the colors bright enough. So these are the reasons why we need to develop technologies that make gene editing simpler, more affordable, and more accessible to patients around the world. And this is something that my laboratory has been working on, getting rid of this complex process and making something that we can actually take to the patient, which can be accomplished in a single office visit inside the body. So we had a recent breakthrough, which I think is probably why the GeekWire folks asked me to come here today. And it really came from just looking at this concept and thinking at a very high level, what are the three major things that we need to be able to do to convert that ex vivo process, outside of the body process, to something like this? First, we need to find a way to deliver these CRISPR pieces, the hand, the scissors, and the code, without electrocuting the cells, which we couldn't do inside of people, and without the need for these engineered virus particles. One step that's passive. If we could do that, then perhaps we could take that delivery technology and focus it to find the blood stem cells inside the body without having to collect them outside of the body. And then if we could show that all of that was safe, we'd really have something that could transform the face of the field. So in the last few years, we've been working on problem number one, passively delivering these components to blood stem cells. And we've done this using nanoparticles. By definition, these are particles that are about a billionth the size of a single grain of table salt. They come in a variety of flavors, protein-based, fat-based, metal-based. I wasn't married to any one of these particular flavors. The reason that we had this breakthrough was actually serendipity. I had a young scientist from Turkey contact me who worked with metal core nanoparticles and was interested in coming to my lab in the US to study whether or not these could have applications in cancer. I convinced him that we should try it in gene editing, and he agreed. What we were able to come up with is this nanoparticle design. It starts with a gold ion core. The first thing that we add to this core is the hand, which is called RNA in this particular system. This couples with the protein, which is the scissors in this particular context. It means that we didn't have to genetically alter or modify or engineer the scissors to get them attached to our delivery system. Now, we had one more thing we needed to add, the custom code or the DNA. But we had a problem. This is very negatively charged, and so is the custom code or the DNA. So like poles of a magnet repel each other, these two things were not going to sit well with each other in solution. So we actually had to add one more component, a positively charged piece that basically attracted that DNA and kept everything wrapped up. This was our three-dimensional design. Here is the actual deep microscope imaging to show that we can assemble these particles in solution. I don't have time to talk to you today, but you can read about it in a scientific publication that came out on Memorial Day last spring. We've been able to show that not only can we make these, but they passively deliver all of those components to blood stem cells, that we do get genetic editing, and we've been able to go on to show that we can correct the sickle cell defect using this approach. So now we've got our passive methods of delivering CRISPR to these blood stem cells. We're working on ways to single out the cells that we specifically want to genetically edit. We're not only working on blood stem cells, but also T cells, as you just heard from Adaptive, but using these to edit the immune system. And we're working on building the models to show that all of this is safe. We just got approval from the National Institute of Health to do an early regulatory gap analysis for the sickle cell disease application. We'll have our first Interact meeting with the FDA in the first quarter of 2020. So this is all incredibly great steps forward, and we're really excited about this potential for progress. But at the same time, this is a great place to take pause and think about the implications of the technology that we've developed. Designer babies are something that researchers around the world have recently tried with CRISPR. This has been all over the news when a Chinese researcher edited fertilized embryos using CRISPR to try and prevent HIV infection. Now, he argued that there was a lifetime potential for these kids to get HIV, but there are so many risks associated with adding these components to those cells that still have to develop into an entire human being, that the risks far outweighed the potential benefits for those kids. Importantly, I want to talk about risk to benefit ratio. We definitely have diseases like sickle cell that are global health burdens, that are very traumatic for the patients that have them, and those are the diseases that we should be going after with simplified technologies such as these. Designer babies are things that the movie Gattaca showed us in 1997 could have massive social implications. Science fiction also picked up on the idea that gene therapy could have consequences well outside of the human space. When Rise of the Planet of the Apes used gene therapy to treat Alzheimer's to create this race of apes that essentially took over the planet, right? And more recently, CRISPR was used to create biological warfare in the movie Rampage. Now, these are dramatic, fictionalized scenarios, but they are things that we have to think about, right? As we make this very simple, very affordable way to genetically edit, not only humans, but other species, we need to be mindful of the applications of those things. And so I started with a computer metaphor, and I'm going to end with one, gatekeeping. These are very simply put systems that control access to the operations or the functions. Each of you, as a custom computer program in this room, is your own gatekeeper. You have the say in whether or not these types of applications should be used in you or people that you love, or maybe you should invest in a company that's trying to use these technologies for things that don't have an appropriate risk to benefit ratio. The rules should be very simple. Alleviate human suffering, and don't restrict access by cost or geography. With that, we can actually make big dents in healthcare burdens around the world, but it's going to take all of us to accomplish that goal. So With that, I want to thank teams of people in clinics who came long before me. I tried to give deference to all of those decades of research and work that happened that brought me to this space right now. Certainly companies that have supported these efforts, people in the community that have reminded me about these things to think about, and especially patients and healthy donors who actually gave their blood stem cells to us to do these studies. So thank you for listening.